Alrighty, welcome to another episode of Learning Genetic Engineering. I am your host, as always, Justin. Welcome to the show. And boy, oh boy, do we have a good one for you today. This is one I've wanted to do for a really long time, and we've had to do two other streams just to get to this point. So I'm pretty excited. Like I say, lots of really fun things. We get to talk about my absolute favorite, actually, that's not true. I was going to say it was my favorite science name, but that's not entirely true. Later in the stream, we're going to be talking about something called the repressilator, which, I mean, sounds just awesome. In hindsight, I realize it's not actually my favorite science name. The repressilator is pretty good, but the skull crucible process sounds better. So, anywho, we're going to get into this pretty quick, but before we do, just a couple of little updates, announcements, that sort of thingy, and then we'll get into it. First things first, as you can see, there is a donation bar at the bottom of the screen here, right there. It says Neuron Supplies. As longtime viewers of the channel will know, we are working on getting neurons to play Doom, because we're mad scientists here. That's what we do. So yeah, any donations are going towards those supplies, so that way we can do the thing. Next month should actually be the next part of the Neuron video, so I hope you're excited, because I'm excited. We've got a whole bunch of really cool stuff planned. We've got all of our culture ingredients en route. So yeah, it's gonna be a fun couple of months. This year is going to be packed with a lot of amazing bio stuff and just a lot of really fun projects. So I'm pretty excited. But I figured that we would start the year off with a stream. Because like I say, I've wanted to do this one for a really long time. So anyway, I hope everybody's doing good. I hope everyone's having a good day. And yeah, let's just get into it, because there's a lot to cover. Alrighty. So, like I said, the theme of the day is genetic circuits. Did I say that? I don't know. I'm forgetful. I'm a professional, I swear. Anyway, the theme of the day is genetic circuits. So in the past couple of episodes of this series, we've sort of talked about the very basics of genetic engineering, whereas what we're talking about today is really quite advanced. Getting into this stuff gets really expensive, because you need a lot of DNA to build a complex circuit, but it's also just really hard. So, yeah, it's yeah, somebody commented, why am I hearing boss music? Yeah, no, pretty much. This is the boss level. And I mean, even then, this is like the intro boss before anything really exciting happens. So anyway, I just got to deal with a couple of things really quick. There we go. I got to just... Okay. Sorry, I just had to adjust our moderator settings. We have a moderator today, just keeping an eye on the chat to make sure that y'all behave, because sometimes people drop by who are a little bit entirely bananas. Okay, we got a couple of donations. Dallins, thank you very much. I'm glad you're excited. Edward Ream, swiggity swooty, I love your content. You give me hope for the future. Aw, thank you. Alrighty, so let's get into it, because like I said, there's a lot to cover, and the sun's fading. Okay, so what are we going to actually cover today? So we're going to start really simple with what is a genetic circuit, because I mean that sounds cool, but it means probably nothing to most of you. So we're going to get into the basics. Then we're going to talk about the basic components, what do you make a genetic circuit out of? How does it work? That kind of thing. We're going to talk about a comparison to digital circuits, because there's a lot of overlap, but there's some very crucial differences. And then we're going to get into some various examples of very basic genetic circuits. So there's a few that we're going to cover, but like I say, these are still the basics, mostly just to give you an idea of what is possible, rather than getting into a specific example of something that's actually really useful to me personally. Because again, I mean, I don't actually do a lot of stuff with genetic circuits like this, insofar as they're just really complicated to get working. So if you can, avoid needing to do it, it certainly saves on cost. But the benefits can dramatically outweigh the costs in some circumstances. All right, so this is sort of the progression of this series thus far. So in the first episode, in the first stream, we talked about single genes. So this is literally the basics. So if we're talking, if genetics is a language, like if learning to program DNA is a language, the first episode was focused on our letters and words, right? So that's a single gene. How does it work? What is a promoter? What is a terminator? How do coding sequences work? What's a stop codon, start codon, Kozak sequences, all that kind of stuff. So we're not going to cover any of that today because we've already talked about it. But like I say, that first episode was the equivalent of learning your letters and words. Then the second episode, we talked about metabolic pathways. So this is the equivalent to like learning a basic sentence, right? So in the case of the example that I have on screen here, so this is the biosynthesis pathway of beta carotene. So starting from geranyl polyphosphate or diphosphate, anyway, GPP or GGPP. Anyway, doesn't matter. The point is, you start with this stuff, you end up with beta carotene. So this is like your basic sentence, right? It's a couple of words strung together that you get a coherent like output. Today we're talking about genetic circuits. So this is where you can take a sentence like this and actually have the organism control like when certain things are turned on and actually be like thinking in a sense about what they're doing and can make adjustments on the fly. So this is the equivalent of like speaking a paragraph, right? We're not writing books here. We're writing a paragraph, but it's still a significant step up from a metabolic pathway. Also, we had a super from 𝐎𝐵𝐻𝖵𝖙𝖎𝖏. Glad you're excited. Thank you very much. Much greatly appreciated. Alrighty. So let's keep. Oh, and we also had one from Randovin Obel. Thank you. Very, very much appreciated. All right, let's get into it. So, basic pieces. What do you make a circuit out of? Let's talk about inducible promoters. So this is actually something that we talked about in the previous streams, but they become the building blocks for much, much more complex circuitry. So what we're looking at here is called the lac operon. So this is a series of genes that exist. Strictly speaking, the versions of this exist in a whole bunch of different organisms, but the one that we're looking at specifically comes from E. coli. Basically the way this works is if this strip here is your lac operon, so things that say 6, 7, and 8 would be distinct proteins that are responsible for parts of the metabolism of lactose. So basically what the E. coli wants to do is it wants to conserve energy, but at the same time it needs to extract energy from food. But it also doesn't want to spend energy on making proteins or enzymes that will break down food that isn't present. So for example, there's no point making enzymes that can break down lactose if there's no lactose present. So the way that the E. coli has solved this problem is with something called the lac repressor. And so you can see this on the left here, and this is literally what it does to the DNA. So basically what it does is it kind of kinks the DNA into a big loop. And you can see that it's got these two ends that sort of target and stick to the DNA at this specific location. And basically what it's trying to do here is just clog up the machinery. So in order for a gene to be expressed, right, in order for protein to be made, the DNA needs to be read. But where the lac repressor binds is sort of in the way. So the place where RNA polymerase would bind to the DNA is sort of before where the repressor binds. So if RNA polymerase tries to stick to the DNA and start transcribing it into RNA, nothing happens because it literally like knocks into the lac repressor and then just pops off because it can't pass it. Like there's this huge loop, there's this giant ass protein, it just cannot get past. However, when lactose is present, it binds to this pocket in the center here and actually makes the whole thing pop off. So when that happens, the loop unkinks and suddenly RNA polymerase can get past with no problem. So you end up with gene expression. So now you have this really simple way of turning a gene on and off based on a signal that came in. In this case, it's a chemical signal of lactose. When lactose is present, the thing pops off, gene turns on. Very simple. So this is like the most basic. This is what everybody uses in like if you're doing any kind of like industrial biochemistry and you want to turn your gene on and off at a specific time, I'd say 90% of the time they're using the lac repressor because it's just so convenient and easy to work with. There's actually a couple of different molecules that'll bind to this pocket, not just lactose. So it makes it just a cheap and easy thing to work with. So a lot of people are very familiar with using the lac repressor. Another one that people are very familiar with is the TET on or TET off system. So the TET on system works exactly the same way. A repressor binds to a piece of DNA, it stops expression until tetracycline is detected, at which point the repressor pops off and gene expression gets going. However, there's also the TET off gene, which instead of it being an inducible promoter that is being triggered by a repressor, it's an inducible promoter that is being triggered by an activator. So it's basically the inverse of this. So instead of a thing that gets turned off when or turned on when a chemical is present, it gets turned. I mean, it still gets turned on, but in the mechanism is backwards. The lac repressor can be a little leaky, but you can double it up. Sorry, I realized I should have clarified. I'm reading a question from the comments here. The lac repressor and some of these repressors can be a little leaky, right? Like we are hoping that this protein stays stuck to the DNA, but I mean it's chemistry in biology, so it's not a perfect system. It will pop off on its own sometimes, or the RNA polymerase can just kind of skip it and keep going, but it does throttle expression pretty significantly. So anyway, the TET on, TET off system is like I say, it's very simple. Either it works when tetracycline is present, the repressor pops off and you get gene expression, or if you're using TET off, when tetracycline is present, the activator binds and starts expression. Wait, did I have that backwards? Sorry, I had that backwards. TET off turns it off when the thing binds. Sorry. It's still a morning. My coffee hasn't hit yet. What I meant to say is TET off turns it off when tetracycline is present. Sorry, that's clarification. TET off turns the gene off. TET on turns the gene on. Both using the same trigger. So you wouldn't want to use this. I mean, you could use both of these versions of the gene. Like if you want to turn, when you add tetracycline, if you want to turn something on and something else off, like this is a way to do that. So yeah, this is another example. And then the final example of an inducible promoter is an actual activator. It was what I'd said TET off was, but this is the actual activator. I knew I'd pick these examples for a reason. I'm just sleepy. Anyway, so this is an activator. So basically what happens, this is the arabinose gene. So when arabinose, which is a type of sugar, is present, it binds the activator and then turns the gene on. And you can see that it does this through like a conformational change, where when arabinose is present, it pops off of this O region, sticks to the I2 region, and that actually turns gene expression on. The specific mechanism of this doesn't really matter, truly. For the purposes that we're going to be using this today, the actual mechanism by which this does this isn't super important, other than how it affects sort of the arrangement of the DNA, but we're not going to really get into that today, because today I mostly want to focus on the mechanics of this rather than getting into the actual nitty gritty of writing the code, mostly just because it's really complicated and convoluted. So I want to just get the basic concepts across first. We got a donation from Dracena. Well, thank you very much. Greatly appreciate it. Okay, so let's move on. Now we have our basic types of inducible. So let's just go over them really quickly again. We have a chemical present, repressor pops off, gene turns on. We have chemical present, repressor pops off, gene turns on. Chemical present, repressor binds, gene turns off. And we have activator, chemical present, gene turns on. So these are all our sort of different flavors of this. I'm sure there are, in fact, I'm certain there are other mechanisms that do the same basic thing, but the important thing to know is things can either turn a gene off or turn a gene on when a chemical is present. Okay, so let's just go over some quick gene notation just so that way you guys know what you're looking at, because we're going to be seeing a lot of this throughout the rest of the stream. So remember, these are the symbols for the different pieces of a gene. So we've got our promoter is always this big sort of fat arrow. The ribosome binding site, which is where the ribosome will bind and then start transcribing RNA into protein, is sort of this like half circle. And then the protein coding sequence is the arrow, and a terminator is just a big T. You'll see that there's two T's because it's just good practice to put two terminators after a gene, just to make sure that it stops. Because I've had issues where as the protein, like as like RNA polymerase is reading the DNA, it'll hit a terminator, ignore it, and then just keep going, and then you end up with a whole bunch of other crap stuck to your protein, which you don't want. So adding the second terminator is always just, good practice. But the important things here are these two lines where you see activator and inverter. So an inverter is basically a thing that'll turn it's the repressor. They say it's an inverter, but it's really a repressor. So when this gene is on, this other one that it interacts with is off. Whereas with an activator, when this gene is on, this other gene that it interacts with is also on. So this passes the signal or turns the signal on. So like if you've got something that is throttling the expression of this, it will then throttle the expression of this positively. Whereas if you've got something that throttles the expression of this, it will throttle the expression of this negatively and reduce expression. Okay, so those are the important bits for the genetic stuff. Now let's have a look at some digital logic. Somebody just asked a question. What about light-inducible promoters? Light-inducible promoters also work the same way. They're light-inducible because there's a protein that's binding to the DNA. So this thing works basically the same way. When the light is present, it pops off gene expression, or vice versa. When light is present, it sticks and whatever. The important thing is that it all works the same, like it's all operating under the same base concept. 𝐎𝐵𝐺𝖆 also thank you for the donation. Greatly appreciated. All right, so let's talk about digital logic, right? So we've been talking about the theme of the day is genetic circuits, but these are the basics of digital circuits. So what we're going to be talking about later on in the stream is how we can implement these sorts of functions genetically instead of with wires and transistors. So let's like if we're just starting at the top here, we have a NOT gate. So what does a NOT gate do? When the signal is high, the output is low. So when you see, I is input. When the input is high, which in digital circuits is a 1, the output is 0. When the input is 0, or low, the output is 1. So this just inverts whatever the input is, right? An AND gate is only going to output high when both inputs are high, right? So if you look, so this is called a truth table. So if you look at the truth table, you'll see that, when A and B are low, output is low. When A is high but B is low, the output is low, blah, blah, etc. When both are high, only then do you get output. You can look at all the rest of these and see the same sort of pattern. NAND is not AND. So it's the exact opposite of an AND gate, right? So where an AND gate is off in all cases except when both things are on, a NAND gate is on except in the case when both inputs are high, at which point then it turns off. An OR gate is looking for either when one input or both inputs is on. A NOR gate is looking for exclusively when there is no input. An exclusive OR will look for inputs exclusively when there is one or the other, but not both. An exclusive NOR is when either they're both off or both on, but not when only one is on. So those are just kind of the list of all the different like digital circuits. Now, how do we actually implement something like this in DNA? Well, it's actually quite easy. So for a simple logic gate with only two inputs, it's actually dramatically easy to program. Oh, hey, Nerd Rage. Thanks for stopping by. Anyway, so to program a NOT gate is literally just the repressor, right? Like the lac repressor, for all intents and purposes, is a NOT gate. Because when input is low, then you get the point. So when the input is low, the output is high, and blah blah. For a NOR gate, what's really interesting is you can actually put multiple repressors before the start of one gene. So if you have two repressors, one after the other, you've essentially built an AND gate, right? Because if you have let's say the LAC repressor and the TET repressor, this gene cannot turn on unless both lactose and tetracycline are present. So now you've got an AND gate. If you want a NAND gate, so you want to invert the output, you have the gene that these two repressors are controlling. When this one is expressed, should repress another gene, so it inverts the output. So for example, if we've got the TET repressor here, the LAC repressor here, and the gene that it's making is the lambda repressor, when both tetracycline and lactose are present, the lambda repressor is going to turn on, and if this gene here is controlled by the lambda repressor, this will now turn off. So let's say this was like GFP, like green fluorescent protein. If you add lactose and tetracycline, you will have darkness. Whereas if you do not have lactose or tetracycline, then you'll get a green output. So it's one of those things where making a very simple like logic gate can be relatively straightforward. Like you don't need to make it, and the nice thing with a genetic circuit is that you don't need to power it. You don't need to deal with some of the stuff that you have to deal with a physical electrical circuit, but you also have like some dramatic downsides, which is you cannot reuse components easily. If you've got the LAC repressor and the TET repressor and the lambda repressor already in use, unless you want another circuit that's inside this organism to also be responding to those same triggers, you can't use the same repressors or activators. So you need to build a catalog of different repressors and activators and stuff if you want to make something more complicated. But you're also going to have to deal with the more stuff you put into the organism, the harder it's going to have to work, because each of these components uses up energy from and material that the cell has available to it. If you're trying to get it to make a specific protein, but you have it controlled by this NAND gate, it's got to, like, the cell has to spend a lot of effort making all of these different proteins just to keep this circuit functional. So you are inherently removing some like material and energy from the cell that it could be using to make your gene of interest. But I mean, that's the cost of this, and the benefits can dramatically outweigh that. And if you design a circuit like this well enough, you can get away with pretty low levels of the repressors, because you don't need, if there's only one or two copies in the cell, you literally need two proteins, like physically, like two copies of the protein for this to basically work. Anyway, so these are some simple examples, right? We've got our basic logic gates. Now, since we can make basic logic gates, the next most reasonable question is, okay, well, can you string them together? Can you do more stuff with them? The answer is yes. I removed an example from the presentation where somebody had built a full adder, which, for those who are familiar with digital circuits, a full adder is the basis of a computer. If you can build a full adder, you can basically build a computer. You just need many hundreds of them. So, I mean, that's inherently limiting when we talk about genetic circuits. But, I mean, it is possible if you are clever enough to do things that complicated. There's also something that I'm not going to talk about today, because it's actually, it's a form of genetic logic that is just so complicated that I couldn't include it today. But instead of using repressible and activatable promoters, it uses recombinases. So these are things that will literally cut chunks of your DNA out and flip them around, or cut them out and stick them somewhere else. So you can do a lot more with that, and you can get a much more complicated circuit at dramatically lower cost, or like material cost, not price. The price is still very high. But in terms of like biological cost, it is much more straightforward. It is, however, way more complicated, and when I was thinking about including it in this presentation, I realized that it was just too much. So we'll end up doing an episode 4 where we'll talk about recombinase memory. So for those interested, that'll be something we'll do in a future episode. But anyway, okay. So let's start with like the most basic of genetic circuits, right? Where one thing represses the other. This is called a bistable switch. So basically, what's going to happen is when you set this up, so you've got a promoter here, which turns on this repressor protein. This repressor protein represses this promoter, whereas this promoter creates a repressor that it represses this promoter. So you can see that it's sort of a cycle. So what's going to happen is if you set this up, like if you make this, and you can see there's an example here where somebody did, what each cell is going to inevitably do is just, like when they first start producing the various proteins, one of them by accident is just going to be produced first. So if repressor 2 is produced first, then repressor 1 just won't be produced, and vice versa. So the bistable switch, when it first forms, will pick a side and then just settle into that side to start with. So it's stable, like that's why it's called a bistable switch. You can make the switch flip states, but it left alone, it'll stay in that state. And so you can see an example here where I think they did this in yeast, and you can see that for the most part, most cells will pick one or the other state and not both. There are definitely examples where you can see that the cell didn't like pick that carefully. And if you look, you can see that like some of these are very white. So if a cell is white, that means it's probably expressing both. And I mean, this comes with the leakiness of this system, and also when you just allow this to like set stabilize on its own, it's not that stable. So like it can flip, or you can end up with like slight expression of both. However, if you've chosen two repressors, so let's say this one is the lac repressor and this is the tet repressor, right? You can add the chemical which interacts with one of the things, and that will flip the state. So if you were to take these, again, I think these are yeast, and you were to apply either lactose or tetracycline, depending on which one you want to do, it will flip the state of the switch. So the cool thing with this particular example is they put the two states with two different fluorescent proteins, just so that way it was very easy to tell when the thing is in each state. But you could, if you were to take this exact same colony of yeast and you were to put them on a plate with lactose, they will turn all the one color, assuming that they were like, these aren't using lactose, these are using something else. But assuming that was what they had built this with, you could have applied lactose to switch the state. And so you can make a bacteria which, for all intents and purposes, will exist under a set of conditions. So like if you just leave some lactose in the media, it will stay in one of the states until you want it to switch, at which point it'll switch. So, you could do all kinds of interesting stuff with this. Like the state 1 could be your protein of interest, and state 2 could be either a reporter, or it could be like an enzyme that does something to either the bacteria or your other protein. So like you could have it build up a huge quantity of one protein, then give it the trigger, and then it'll only focus on like post-processing, for example, depending on how you write this. But this basic format can be used for whatever your purpose might be. So yeah, it's a nice, really basic circuit, but even with just two repressors, you can get like really interesting behavior out of something which is biological. Like you get this really distinct bistable state, which is very weird for bacteria. So just really quickly, I see a comment. How far are we from DNA memory storage for computers? The recombinase memory that I mentioned a moment ago, that is what that is. And I've seen some very, very, very complicated examples, which is why I wanted to save it for another stream. Because if you're using DNA memory, it gets crazy. But DNA memory is also iffy because DNA is not a very stable molecule. It makes a lot of mistakes. So, you've got to have a lot of error correction, but you got to have error correction on a bacteria, which is just weird. But still. Alrighty. So yeah, bistable switch, nice simple example, works really well. Lovely. Okay, now we get to talk about my favorite. This is when I first saw this, like I wanted to do this stream like the next day and do nothing but the repressilator, but I realized there just wasn't enough on its own. But yeah, so this is called the repressilator. I forget the authors who first figured this out. I'll put that in the description. Also, I will say all the stuff that I'm showing today, I'm going to put a bunch of links after the presentation into the description of all the links to all the different stuff where all this is from, so that if you're interested, you can check it out. But anyway, so the repressilator is basically one step further than the bistable switch. So instead of it being two things that repress like each other, it's three that repress in a cycle. So what's really interesting about this is you can, like, this goes from a thing that has two stable states to something that will oscillate infinitely. As long as you have an odd number of repressors, you will always have oscillation. If you have an even number, it will settle into a stable state. So if you've got 4, you'll basically have a quad stable switch where you can have it switch between 4 modes at different strengths. Whereas if you have 3, 5, 7, whatever, you'll always have oscillations. And what's really interesting about this is you can actually pick off the output from each of these repressors. So if you have the same promoter drive, so let's say you have 2 plasmids, and actually I can just show you what this looks like. So here's 2 plasmids, right? Where it's the exact same thing we were just talking about. It's 3 repressors in a row. So we've got the TET repressor, the LAC repressor, and the lambda repressor from the lambda-phage of it's a virus that infects bacteria. So basically we've got 3 repressors in a row, right? This one represses this one represses this one, and this one represses this one. So it will always cycle. So you'll have one thing build up for a while, and then as it builds up and builds up and builds up, the levels of one of the other ones drops, which allows expression of the next one, which then drops the level of the one that was building up, et cetera, et cetera. So what they've done with this is they've built 2 plasmids. So we have our main repressor cycle here, but then we have something that actually picks off the input. So in this case, they've got the TET repressor, right? And the promoter for the TET repressor is driving both the lambda repressor and GFP. So what's really nice about this is whenever the TET repressor is active in the cycle, GFP turns off. So you in a colony of bacteria, you will end up with periodic patterning where basically certain bacteria will be producing certain fluorescent proteins based on which repressor is expressing. So in this particular example, they've built a triple. So you've got 3 repressors and 3 reporters. So that way you can actually visually see what the bacterial colony is doing at each step. And you end up with this amazing periodic patterning of 3 different colored fluorescent proteins, but in a single colony of bacteria. And if you actually grow this colony in a tube, you'll literally, and the tube is actively mixing while it's growing, you'll see the entire tube change color. So if you get a time lapse of this, you'll see it go from green to blue to red to green to blue to red over and over and over again, at least for a couple of cycles before the the oscillations, they're not perfectly in time. They're relatively well timed, but it's still you'll get a little bit of drift. And so after a little bit of time, because you can actually see it in these rings, right? Do you see how there's these little bits that boop out and aren't perfectly round? That's because these bacteria are a little bit further ahead in their particular cycle than the ones right next to them. And so these errors will build up, and eventually you'll lose the coherence between all of the different like all the cells in the colony. So if you've got this in liquid, you will eventually see the color changing stop. But for the first couple of cycles, you will absolutely see it color cycle, which is why I want a sample of this desperately. I've been meaning to have this made for a while because I want to show it on the channel and I want to do a full video where we do stuff with this. But it's a humongous piece of DNA because you have to have all of this repressor stuff and you have to have a whole second thing with all the fluorescent proteins. So it's just a huge print. Yeah. But yeah, so Nerd just mentioned I want color oscillating fish from my aquarium. Legitimately, this is something you could do. Like, and we're going to talk about this, but cycles like this exist in nature already. The way things get their patterns and colors, like if you're looking like at a snake or a reptile or a fish or whatever, the way that it got those patterns is because of natural oscillators like this that are built into the fish or snake or whatever. And you can actually go in, figure out what that cycle is, and then mess with it and either adjust the timing or adjust the output or adjust how it works. But we'll get to that example as we get through more of the presentation. So here's another example of the repressolator where you can see the cycling more specifically. Here's one I found on Twitter from somebody who had done this, David Rilger, I believe, and his lab had made a version of this. And I mean, I just love this oscillating pattern because these are like bacterial colonies. It's so weird to see a bacterial colony that has patterning like this built in. Over here, you can see another version of the repressolator where they did the 3 colors. Basically what they've done is each of these images, they're looking at it through a different emission and excitation filter. So in this case, they're shining a sort of UV light on it and looking at it with a blue filter. So you can see the blue fluorescent protein, and then the same thing with the yellow and the purple. But they chose these colors so that way they're as visually distinct as possible. But if you look really carefully, you can actually see that right in the center, and I don't know how well this is going to come across on the screen, but right in the center, there's actually a bright spot in the blue, whereas at the same time point, there is no bright spot in the yellow, but there's a very big bright spot in the pink. So you can actually see where the cycle is happening, and you can see that these rings there's not the same number of rings in each of these pictures, right? There's 3 in the pink. There's, there's 3 or 2.5 in the pink. There's like 3 good ones in the yellow, and there's 4 in the blue. So you can see that like the blue must be the first in the cycle, so it's evidently expressed more times, whereas pink must be in the end of the cycle. And I think BF is just like white light, so unfortunately, like it doesn't necessarily look this. It's either that or this is a control. You do need the fluorescent lights to actually be able to see this. But I do want to try this and see if we can get, because I mean, one of the problems that tends to happen in papers like this is they choose fluorescent proteins that just aren't very bright or aren't very colorful, or the expression isn't very high. One of the problems with this sort of system where you're just using a repressor to turn something off is that if the actual promoter like filters, whereas there's other ways to increase this thing. So for example, if you were to actually build a NOT gate into one of these, so like if instead of just taking the raw output from one of these repressors, but you invert it, you can end up with a much stronger gene expression. Output. So if you've got a promoter that is very weak, but triggerable. So like, for example, there are some... Are we good? Is my audio fixed? Oh, shit. Okay. I think I yanked the cable of my mic. Sorry, guys. All right. Okay. We're back. We're back. I'm alive. Is this... Okay, is it working now? I'm gonna just keep talking. Surely this should be fine. I know what happened. I think I yanked on the cable. My mic is a little bit fucked. I had to use a weird cable today, and it's kind of short, so I think I just bumped it. Yeah, the problem is I also gesture with my hands a lot when I talk, so I think I just yoinked the thing. All right, anyway, we're back to it. So what I was saying before was if you use an inverter, you can get higher expression. So there are some light activatable promoters where the expression from that promoter isn't very strong, but you can use that in an inverter. So if you've got a light activated promoter and it's driving expression of a repressor, then you can have that, or an activator actually would be a better example. So if you've got light activated promoter driving an activator, and then that activator turns on another gene, you're going to end up with much higher expression than the light activated promoter. So yeah. Okay, let's keep moving. So yeah, we've looked at this, and yeah, this is what I was talking about before. If you've got an odd number of nodes, you'll always have oscillation, whereas if you have an even number, you'll just hit these like stable points. And then you can shift the cycle by applying the chemical or whatever that these interact with, but it will settle into a solid state. Somebody's asking, a new cable can't be that expensive. Well, I mean, donate, I'll buy one. But yeah, anyway. So yeah, that's the repressilator. It is one of my favorite genetic circuits, but just because it's a hilariously named thing, but it's also just really cool. And like I say, this is sort of how the plasmid would look. So you've got, in this case, they did it as two plasmids, but you can see that because they did it that way, they need two different antibiotic genes because you've got to apply both antibiotics in order to keep both plasmids in the bacteria, which is just not like a good plan. I hate designs that do this. Just put all your stuff on one plasmid or integrate one of them. Like, it's just silly. But anyway, so, you've got your origin replication, your AMP resistance, but then you've got your three repressors, and then you've got your pickup plasmid that will put out your reporters. And again, you can have as many reporters and repressors as you want if you can find them. Somebody asked, why does this lead to oscillation? So it leads to oscillation because these are all repressors, right? Like this one turns off this one, which turns off this one, which turns off this one. If this one is on, this one is off. But if this one is off, this one is on. And if this one is on, well, this one is off. You see what I mean? So like it just goes around and around and around in a circle. And yeah, somebody said, would an even number work? No, it wouldn't. An even number will stabilize. So yeah. Anyway, let's keep moving. Okay, so now we've looked at the repressilator where all of this function is happening inside of one cell, right? Like there's no communicate. Like the amazing thing about this system is that you get this periodic oscillation and patterning even though there's no communication between the cells. It's just that reliable. Like the oscillations are just that coherent that you end up with this effect whether or not there's any communication between the cells. However, what happens if you have an element that leaves the cell and then another cell can pick up as a signal? Now we start getting into really interesting stuff where you can pattern not just inside your the one cell, but also at a distance. And you can start sending signals between cells and build more complicated circuits. So, there was one paper that we're not going to go through today, but I'll tell you about briefly, which they used diffusible elements, and you're able to do all kinds of different patterning stuff. And they basically had combinations of cells where each different strain had a different circuit in it, but also had the ability to send out chemical signals which the other circuits could pick up. So basically you can build a very complicated system. So this was one of the ones that built a full adder. So they built a full adder with all of these different logic gates, but the logic gates were contained in physically different cells that were passing signals between each other to communicate, which gets really, really wild. But so let's go through what happens when we start adding diffusible elements, right? So this is a level 1, or what this paper called a level 1 diffuser. So this is one diffusible element, so the D in this situation. So we can look at this more specifically. So this is this example. This is the level 1 diffuser. And this is basically how this works. So you have two different circuits that are running at the same time. So you have the sender circuit and you have the band detector circuit. So basically the way this works is when you apply tetracycline, the 𝐴𝐷𝖆 gene turns on, and this produces a chemical called AHL. AHL, I believe, is acyl homoserine lactone, or just AHL. And it's a chemical that readily diffuses outside of E. coli. And so, but it is also triggers gene expression. So it's an activator, but it's an activator that leaves the cell. So you've got one gene that physically manufactures the chemical, which then leaves the cell, and then you've got another circuit which will pick up that chemical and then do something with it. And so the way that they set this circuit up is actually really interesting because they had programmed it such that only a specific level of AHL will actually turn the circuit on. So for example, when there's very high levels of AHL, so the way that they did this is they've got like a lawn of bacteria on this plate, and then they put a little piece of filter paper in the middle with tetracycline on it. So only cells right near this zone of the paper is going to have tetracycline, and which means only these cells, the green cells in this case, are going to be sender cells. So they actually have enough tetracycline that the 𝐴𝐷𝖆 gene turns on. And then they start to exude the AHL. So cells near them will actually start turning on. So when you've got this like gradient of AHL concentration, the way the circuit is set up, when it's very, very high, it will activate two repressors. So the LAC repressor, version M1, and the lambda repressor, which is CL or CI. So when LuxR is very strongly activated, both of these turn on, which very strongly represses both of these other genes. When AHL levels start to drop, this activator doesn't work as well. So the first thing that it's going to do is this lambda repressor might still be working, but the LAC repressor, which is leakier, won't. So if the lambda repressor is on, this LAC repressor is off because it's an inverter, right? Whereas this LAC repressor, if it's off, then the GFP here, the reporter, is on. And then there's also a further repressor here. So this is like an on, then a not, and then a not. So when you've got two knots, it can pass the signal. So when there's really, really, really low AHL, LuxR is off, LAC1 is off, CL is off, but this other LAC is on. So you get no expression. When there's a medium amount, you get expression, and when there's a high amount, you get no expression. And so you end up with this really interesting diffuse pattern sort of structure where you can get expression at two different places and pattern that way. Of course, in this case, because you're relying on just sort of a sweet spot of AHL, you can't get a repeating pattern unless you had another source of tetracycline. And in this paper, I didn't include the image just because it didn't fit, but they did another experiment where they just put a couple little discs of tetracycline, and you do get a more complicated pattern where you still see that like zone of inhibition, but then you also see around the periphery a zone of glow, which I think is really cool. But yeah, so this is a single diffusible element where the diffusible element has to leave the cell and go to another cell to do something. So now this is a self-interacting one. So this is what the level 2 diffuser here, where the diffuser leaves, but also represses itself. So the way that this one works is AHL is the chemical that has to diffuse out, right? But there's an enzyme, AIIA, that, there's an enzyme, AIIA, that breaks down AHL. So the higher the level of AIIA there is, the lower the amount of AHL will be. But the way they've got this set up is that the promoter that is driving both AIIA, the production of the AHL, and a fluorescent reporter are all driven by a promoter that is turned on by AHL. So when AHL is high, you will both have expression of the AHL synthase gene, you'll have expression of GFP, and you'll start getting expression of AIIA. But as AIIA builds up, it starts breaking down the AHL, which turns off the gene. And so then the enzyme that's breaking it down turns off, but then the level will start to build up again, and so the thing turns on, and then you're, on and on and on again. The reason this one works is because the bacteria naturally make this, so you'll always have something to kind of turn it back on. Because this is one of the chemicals that bacteria use to do quorum sensing and communicate and that kind of thing. So it's, But so the way that they did this experiment is they built this sort of microfluidic trapping chamber. So you've got a population of cells stuck in the chamber, but as they grow and push on each other, they end up in this channel and they get washed away. So this way you can basically see a constant flux of the bacteria as they change over time. So you can see that over time, and it takes, 15 to 30 minutes for it to really build up. As the chemical builds up, you start getting this really bright expression until it then starts getting broken down again, and then the expression turns off. But then you get this periodic cycling where it goes high and then low, and then high and then low, and then high and then low over about an hour. So, it starts low, and then it sort of peaks at about half an hour, and then by an hour, it's starting to slow down to almost nothing. By like 75 minutes, it's basically at nothing. And then it just does this over and over and over and over and over again, which I think is fascinating. All right, so we're making good progress. Okay, so let's look at a couple of examples and some natural examples, which will also show sort of like, kind of what you can do with this, but also some examples from nature. I will say I couldn't find a good example of the level 3 diffuser, and what they're showing here is actually not real. This is like a simulation. However, if you have two diffusible elements where one interferes and one activates, and one self-interferes as well, or self-activates. So yeah, this one self-activates, but it activates this other one, and then this one turns it off. If you've got something like this, then you end up with these really amazing patterns. And when you look at this, this should become very obvious why you start seeing patterns like this showing up in biology. Because in, say, a snake, or a fish, or whatever, like you see these sorts of patterns because you have these interacting and self-interacting cycles where you end up with these like high and low areas of expression. And then if you've got reporters like picking off data from this cycle, then you can have these really cool patterns. I want to build something like this really badly. I think that it's going to be really cool looking. Because if you made a lawn of bacteria, it doesn't actually matter where the bacteria are. They will settle into these patterns just because they're like sharing the chemicals back and forth and back and forth and back and forth. So you can get some really amazing patterns, which if you are, and it doesn't need to be fluorescent, right? Like these patterns could be driving something that is glowing. It could be something that is a pigment rather than a fluorescent protein. So you could be making melanin or eumelanin or biliverdin or whatever the chemical is. The nice thing is once you have these cycles established, you can pick off the output from any point in the cycle to do something with, with whatever it is that you're trying to do. Like I said, I'm going to put references and links to all of the different papers that I used for this presentation in the description after this. I just kind of ran out of time today, so I'm going to do it after. But yeah, that'll all be in the description for after this. Okay, so let's talk about some examples. So this is like the really classic example where like the circuit doesn't need to be very big in order to do something interesting, right? Like this is a very, very, very simple circuit where it could be looking for, let's say, 3 things. So it's looking for, or actually, I guess this is 5 things. This is like disease markers. This is drugs and quorum and location. So location would probably be pH, because this is depicting intestinal lining. So if you're going for location, you're going to want something that's probably pH sensitive, because you want something when the pH is low that it turns on, because you don't want this turning on in your stomach, for example. Or you could have this temperature dependent. So if this leaves your body, like if it drops below a certain temperature, you can have this like self-destruct and just die. So this way you can have control over what happens to your organism after applying it in practice. If this bacteria is also able to make a drug, you can have it be location dependent. So you're looking for when it's, say, in your intestines. Then it's also, then when it's, got the correct location, and it's detecting a disease marker. So let's say you have Crohn's or something, or celiac, or something that like, or inflammatory bowel disease, or something that makes like a sore or like a cancer or something. You could have the bacteria be sensitive to a chemical that the disease releases. So if it's like a sore, you might be looking for like calprotectin. You could be looking for, maybe it's you're looking for the quorum sensing chemical from another bacteria or something. When that is detected and it knows that it's in the right location, then you might have a drug produced where it could do something like produce a drug but also be monitoring itself. So when it detects that the level of drug is sufficiently high and quorum sensing is sufficiently high, so that means there's physically the level of bacteria passes a point, you could basically have the cell self-destruct. So it can either be a self-destruct when it leaves the body. So like if it's a temperature dependent self-destruct, when you poop it out, it drops in temperature, and so the bacteria explode. Because you can program, this says CRISPR-RI, and so this just stops replication, but I saw a really clever way to do this where it uses what's called a meganuclease. So it's basically an enzyme that just shreds DNA, like all of it. So you basically have this circuit where it's very sensitive to its outside environment, and when it detects that it's either done its job or it's left the area that it's supposed to be in, it triggers the self-destruct, and it basically shreds all of its own DNA, effectively killing the bacteria. Because if all the DNA is just mush, it doesn't work anymore. Like the only reason DNA works is because the code is ordered and precise. If you turn it into just soup, it stops working. So in this way, you can build biosafety into a highly engineered system like this. Alternatively, you could have this be, instead of a bacteria, this could be a virus. So let's say that you're trying to do genetic engineering in a patient, but you really only want something to happen in a very specific location. So like this could be a cancer treatment. So it could be a virus that you apply globally, and I don't mean globally like to the world, I mean globally to a body. So the virus just goes in, gets into all the cells that it can, but then the circuit is looking for the presence of specific things in those cells, and will otherwise just self-destruct, not necessarily the whole cell, but you can set it to self-destruct only itself. So, for example, like you could have it check to see if a tumor or like an oncogenic gene that you know is fucked is present in high levels. And if it is, then you could have the circuit turn on and produce something that kills that cell. Or if it doesn't detect that any of the tumors or oncogenes that you're looking for is present, you can basically have the virus self-destruct. So that way, if it's not where it's supposed to be, it leaves the cell alone. Whereas if it is where it's supposed to be, it becomes like a tiny bomb, and just kills the cell and the whatever the case may be it a cancer or a tuberculosis or whatever. But I mean, like you could do this with like tuberculosis, for example, where you could infect the person with a virus that if it detects a cell with tuberculosis in it, it immediately shreds it. So instead of having to need new drugs for killing tuberculosis, one thing that you could do, like if you really wanted to get into the weeds with this, you could have a circuit which is looking for the presence of tuberculosis inside of a cell, and you deliver this with a virus, so that way it can just get into whatever cell it needs. When that virus it enters into the cell, the circuit boots, and what the circuit will be looking for is, let's say, it's looking for tuberculosis. If this happens and it detects the tuberculosis cell, you could then have that virus turn on and produce something like a bacteriophage that is specific to tuberculosis. Because one of the interesting problems with tuberculosis is that, unlike most bacteria, which tend to hang out outside of your cells and will just like kill cells and then eat the goo, tuberculosis actually like gets inside your cells and then hides, which is why it's so difficult to treat. And this also makes it a problem if you want to treat it with a phage, because the phage has evolved to get into tuberculosis cells. It hasn't evolved to get into human cells. So the only way a phage can kill tuberculosis is if it gets into the cell that the tuberculosis is hiding in, but how do you get it in there in the first place? And so some people will use, or some papers have used another bacteria that can technically infect you, but like it's not infectious. If that makes any sense, like it can get into your cells, but it's not damaging. And so they'd use that to deliver the phage, but like that's not really an effective way of doing that. Whereas something like this, where you've got a virus with a control circuit in it, it just goes everywhere and then checks if it's in the correct spot. If it isn't, it like self-destructs, and if it is, it does its job. And this could be the same where, I mean, it's bacteria, right? Like these are little chemical factories. They can make anything you want if you are sufficiently like dedicated. So, you could have a virus that looks for tuberculosis in cells and then produces the antibiotic that kills them. But only locally. So that way the amount of antibiotic that you actually need is unbelievably low. But to the cells that are affected, the concentration is going to be extremely high because it's being produced by the cell that's affected. Or you can produce a chemical that interferes with tuberculosis or becomes an alert. Like you don't even need to kill the tuberculosis necessarily. You just need to tell the body that that's where it is. So if you were to make a circuit that is looking for the presence of the tuberculosis, then you could just have it produce something very irritating to the immune system. So there are all sorts of proteins that are well known to piss off the immune system. So if you just have a circuit that is looking for the presence of tuberculosis, and then immediately when it detects it starts producing something irritating, the immune system will rush to that area and just kill that cell. And whether or not it knew the tuberculosis was there becomes a moot point because it's all dead. So you can build really, really, really complicated examples of this that can do all kinds of amazing and interesting things. But like your circuit itself doesn't need to be that complicated to have an amazing or interesting effect. And, the same can be applied to like biotechnology where you're doing like massive protein synthesis. Like you can have your bacteria or yeast or whatever follow a program and do things and be checking their own progress to know when to turn certain states on and off. This kind of thing. Somebody's like, you talk about immune system as it has sentience. It doesn't have sentience. It follows specific rules. But if you know what the rules are, you know how to make it work. So like the immune system, when I say it pisses off the immune system, like the immune system is constantly looking for things that are not you, right? So like, for example, a great protein that would be a phenomenal thing that would definitely alert the immune system to there being a problem is, say, like COVID spike protein. So for example, you could make a circuit that looks for, I'm just going to keep using the tuberculosis example because I watch a lot of John Green and it's in my head, so just deal. In the tuberculosis example, right? If you apply the virus that detects the tuberculosis, when it does that, it immediately starts producing COVID spike protein. If that were to happen, every single cell that is producing that spike protein would get stomped by the immune system very, very, very quickly. Because, I mean, as long as you're up to date on your vaccines, the body knows what spike protein looks like and how to kill it. And every cell is supposed to do what's called antigen presentation. This can get affected in infected cells and in cancer cells, but what cells are supposed to do is every protein they make, at least in a human or mammalian context, every protein they make, they take little pieces of it and sort of pepper the outside of their cell with these little pieces of protein, specifically so that a specific type of immune cell can go and bump into and like lick every single cell that it passes. And if at any point it detects something that's not supposed to be there, it immediately causes an alert that alerts the whole rest of the immune system that something's wrong. So if it detects something that's not supposed to be there, it starts releasing cytokines and all kinds of other alert molecules, which will attract other immune cells to the area. And so you'll get something like a, whatchamacallit, like a natural killer cell to come in and just like stomp that cell and just eat it and kill it. And that's the end of it. Of course, this can be affected by whatever disease it is you're trying to treat. But I mean, you can build in stuff like this. So like if TB were to inhibit, antigen presentation, for example, I mean, you can just hijack it. Like the nice thing with applying a virus is like, yeah, no, it can inhibit antigen presentation all at once. But like if you code it correctly, you can have a thing that is going to pepper the outside of your cell with spike protein. Right? Like you can engineer, like if you take a like a normal plasma membrane bound cell, or not cell, a plasma membrane bound protein, something that is always on the outside of the cell and is always trafficked properly, and then you stick a spike protein to the end of the protein that is outside of the cell, it doesn't matter if antigen presentation is turned off because the cell has a giant spike protein strapped to the outside of it, and then it narcs really hard. But this is it's one of the like the nice thing is like diseases are only a problem if you can't just grab it by the DNA and squeeze. But like if you can, it doesn't matter what defenses the thing has made. You can just subvert them. Like it's like, oh yeah, it reduces expression of the thing that tells your immune. Okay, fine, so crank it. Like if your virus has a really, really, really strong promoter in it, it doesn't matter if the thing is getting stomped. You can overwhelm it. So like there's lots of ways to get over all of this kind of stuff. And the nice thing with genetic engineering is that if you are sufficiently motivated, there is essentially nothing you can't do. It's just really hard and expensive. But yeah, no, I mean, it's one of those things, like if you ever look at the list of incurable diseases, the only reason they're incurable is because we're not allowed. Like we do actually have treatments for most of them. Or we know what would absolutely fix it. It's just, they don't let us. So like if it's a genetic problem, you're like, all right, well go and fix the gene. They're like, no, that's gene therapy. That's highly regulated. And you're like, but why? We can just fix it. This is silly. But anyway, that's a whole separate thing. Anyway, let's move on a little bit. Okay, so these were some examples of like what humans can do. Let's have a look at what nature can do, because nature's way better at this than we are. So like keep in mind, this is like 6 little activators and then a little repressor there, and like 2 little cycles, nothing big, right? Okay, this is control of flowering in Arabidopsis. It's mildly more complicated. So these are all of the different things that the plant is looking for in order to know when it's supposed to make a flower. And this doesn't even count like you see this arrow that just says flowering? Like this doesn't even, oh, actually, you guess you can't because it's behind the thing. There, do you see that? It says flowering underneath it. Go ahead and I'm gonna shrink this bar a little bit so that way. Shrink. Okay. There we go. Now you can see flowering. Okay, so yeah, flowering is an even more complicated circuit than this, but like this is just the control circuitry, right? Literally, this is just the control circuitry for when it should flower. And really, the only thing that needs to happen is FT and FD needs to activate this stuff, like needs to turn on, basically, and then it just turns all this shit on. But look at all the different things that will either increase or decrease the expression of this. Right? Like if the vernalization pathway hasn't been, like if this program hasn't run, vernalization, by the way, is the process of putting a plant in a really cold spot. So like, how some plants need a cold cycle in order to flower or grow the next season? Like that's what vernalization is. So there's this whole pathway that if this program has not run, this one will not run. So, you've got to actually like do all of this. It's a freaking nightmare. But I mean, it's one of those things where if you wanted to turn vernalization off in a plant and you knew that all you got to do is break this one gene, I mean, you could do that and then it'll just be flowering constantly as long as all the rest of the circuitry is also correct. So like if you just want to get rid of the need to freeze the thing, So then, you could do that. Like, for example, amaryllis or like tulips. Like there's a reason that like most bulb things need to be kept in the fridge or freezer or go through a winter cycle in order to bloom. It's because they've got this vernalization code built in. So you can go in and start messing with the circuit and doing this. But like one of the things that I find really interesting is that when you look at different morphs of the same species, so for example, like ball pythons, right? If you're looking at different morphs of the ball python, it's all the same species, but like the way that the code has been activated and interacted with changes between the morphs. But behind me, you can't really see it because I got my whiteboards up, but wait, actually, if I just go to just camera, yeah, you can kind of see it like right here. These are some like behind the whiteboard is a bunch of plants. I've got a bunch of different varieties of pepper, for example. When you look at each of the different varieties, you can tell that they're all the same. You can tell that they're all the same species, but the way that their genetic circuitry is wired, you can see the differences. So for example, like the petiole on one is dramatically longer, or like the leaf is a little bit wider. And it's all because something in the genetic sequence and in its circuitry has changed. So, let's say like one of these might run for a little longer, or its repression is a little bit stronger. This kind of thing. So, this is just control of flowering, just to show you how complicated some of these cycles can get in nature. And this is like a fraction of the whole diagram. Like when you actually look at the whole control circuit, it's just bananas. And it's taken people decades to figure all of this out because, I mean, look at this mess. This is like whenever somebody's like, biology is perfect, we've made by God. I'm like, bro, have you seen the code? Like this shit's held together with like duct tape and zip ties and it's chaos. You're like, yeah, why is it like this? I don't know, it evolved that way. It basically works. Anyway. That's control of flowering, just to give you an idea of how complex this can get. But now we're going to get on to our last example, which is embryology. So this is an example of one of those natural clocks that I was talking about. So, when you've got the repressilator, you can get this periodic motion, but there's all sorts of... What happened? But there's all sorts of different clock circuits that are running in any particular organism. Right? So what's really interesting is this is the embryology of snakes, and this is actually going to be the subject of our next stream. So the plan for the streams for this year, today was going to be programming genetic circuits. Next time is going to be making snake people, and then the last one is going to be making sandworms from Dune. So I had to do it in this order, otherwise they're not going to make sense. Today's circuits, next time snake people, then we make the sandworms. So anyway, there's a genetic circuit going on in snakes and also in every organism, like as part of the spine development. And what's weird about snakes, and the reason why snakes are so long, and the fact that they're basically just a spine, is because their clock runs faster. So for the same amount of growth time, they produce dramatically more somites, which eventually become vertebra, than, say, a lizard. And the reason is that their clock literally ticks faster. So something has changed their circuit that makes it tick faster, and that alone is enough to get a dramatically longer spine. So like if you take, for example, this clock, like if you take the clock from snake and apply it to mouse, you will absolutely get a snake mouse. And like if you get rid of there's a single gene that you can mess with that'll get rid of the limbs, and then you'll literally have a snake mouse. So like it's a matter of like a couple of genes, and suddenly you have all kinds of interesting stuff. Also, you can like pick off output from other biological processes for your genetic circuits. So for example, instead of using if we go back to this example, right? Like instead of using like a temperature-based location, if your location signal was coming from a Hox protein, you can localize expression to a single body segment if you want. For example, like if you were to choose, I believe Hox, I want to say it's 3. I'm gonna have to double check this, but like, it would be sort of the like torso-y area. Whereas if like Hox 1 would be more like head. So, you could confine expression, like you could have that oscillating circuit, but only run like for this little body segment right here, or like only in the tail. So if you wanted, you could pick off the output that says this is tail, and then have this run a clock that then periodically makes color, for example. So you could like get patterning that way. But it's really just limited by your creativity at this point. Like once you have the ability to make complex circuits like this, and also to analyze the circuits that already exist, you can pick off output and then redirect the gene flow however you want. Of course, there are limits. You'll break the fucking thing at some point, but within reason, there's very little you couldn't do. Yeah, so that's pretty much it for genetic circuits. So from here, we're going to kind of move on to questions. I know that our moderator has been collecting questions throughout the whole stream. Yeah, if you've got questions, comments, now is the time. So I'm going to set this back to just camera. Alrighty. Our moderator should be sending some questions. So let's see what we got. Bibbidi bobbidi, where's my questions? Questions? Bro, I need some questions. All right, anyway, I'm just going to start taking questions from the comments until I get my... Oh, there we go. Yay, I got my list. Okay, so somebody asked, how would the speed of DNA computing stack up against conventional computing methods? Terribly. Biology does nothing fast. Like, truly, there's the only thing that's fast in biology is things that run. Like, and even then, that's not like, yeah, no, nothing is fast about biology. Like a digital computer is going to be exponentially faster than any kind of DNA circuitry. The only thing that's even relatively fast in biology is like neural computing. But that's like a whole other thing, and that's really, really complicated. Okay, what else we got? Somebody's like, I wonder if we could make a diabetic fluorescent indicator that could be injected into skin cells. Yeah, no, absolutely. You like 100%. That would be very straightforward to make. If you wanted to have like a reporter, like a DNA tattoo reporter. The trick is you would have to have something that'll break down the protein after the alert. So you could have a patch of skin that like turns green, when you're in need of insulin or whatever. But the problem would be getting rid of that GFP when it's not necessary, and also, or rather, once the signal has passed, and then also making sure that none of the genetic circuit gets stomped by your immune system. So one of the things that I didn't mention is like all the things with genetic circuits, if you're going to apply it to an animal, either has to be applied before the thing is born, or you expect that it's going to fade very quickly as the immune system stomps on it. Otherwise, you have to actually purposely build in mechanisms to basically calm the immune system down and make sure that it doesn't attack the cells carrying the thing. So, yeah, that's the thing. Okay. We just got a question from Tatiana asking thoughts on using enzymes to recycle plastics. I'm gonna burst what I assume is a few people's bubble here. Plastic is not recyclable. Plastic should be burned or stuck in the ground. But like plastic is not recyclable. It's like the lie of plastic manufacturers is that they say that it's recyclable. It takes dramatically more energy to recycle plastic, and also almost all of it that it says is getting recycled is just going into a landfill. So you can't actually recycle plastic. It makes way more and also plastic only accounts for about 1% of the like carbon emissions and production of the world. So like if we were to just burn all of our plastic, like in a really good incinerator to make sure that it's not just making toxic garbage, and then doing carbon capture, like that's how you recycle plastic. You can't actually it's just the problem is there's so many different kinds of plastic and you cannot mix them. So you have to have perfect sorting, which just becomes unreasonable. Like there is a couple of countries that have attempted it, but what they don't say is that they're actually like throwing a lot of it into a landfill and they're just not telling you. So yeah. Okay, let's moving on. Okay, if I make a plant produce luciferase, will its seeds spread the gene? Yes, depending on how you code it. That is a thing that is very dependent on how you code the thing. Um, one of the problems with plant engineering is if you're not careful, the gene can not just spread to the offspring, but it can actually spread to the wild. Because if you don't code the gene properly, it'll end up in pollen, and pollen from different species can like do horizontal gene transfer. So you can end up with like a weed picking up like your Roundup Ready gene, like your glyphosate resistance, which is bad, and because it just ruins the whole thing. So you've got to like really carefully design your stuff to like limit it to like chloroplasts, for example, because those don't end up in pollen. But yeah, no, I mean, if you design your thing and it's like integrated into the host genome, yeah, it'll pass. Okay, what else? Does it make sense to express the genetic logic as NAND logic like you would in silicon? You can, like, you can make a full adder with genetic logic. It's just really complicated because you need so many different elements. So it's tricky, but it's doable. Is there any estimation of the efficiency to power a genetically driven computer versus an electronic one? Would this be more cost efficient if it was scaled up? No, not really. I mean, biology is it's efficient, but it has very limited complexity when we're talking about genetic circuits. So like it just doesn't scale. Whereas like transistors scale really well because you can make them literally like 1 nanometer or 5 nanometers wide, whereas like a genetic and you can use the exact same topology over and over and over and over again. Whereas in genetic circuits, you're very limited because you can do like 3 things and then you run out of pieces. Okay, what else we got? How far are we from DNA memory storage for computers? For computers, forever. It'll never happen. It'll never happen. It's a terrible, terrible, terrible idea to actually use DNA for memory storage. DNA is not a stable molecule. I don't know why people keep trying to do this. It's so stupid. Like, DNA is not a stable molecule. The whole fucking thing with DNA is that it is constantly getting changed because the polymerases make errors, and also things just break your DNA all the time. So like it's always going to drift. Of course there's like limits, but at the same time, like, it's just so delicate. Like, you know, if you keep it wet, it'll just break down on its own with time. And like DNA is self-enzymatic, like it can fold into enzymes, so it can cut itself apart. And like there's transposons, which will cut themselves apart. That's part of how they function. So like it's just not a good plan. Like, again, you can get transistors down to like nanometers, like single-digit nanometers. Like there's no reason to do it with DNA. It doesn't make any sense. It's also just not stable. It's like nobody wants unstable memory because then your computer's like, oh yeah, why did it die? I sneezed in it. Like, what the fuck? Anyway. Okay. What else we got? How do you come up with this stuff? It's honestly mind-boggling. I just read a lot. I read a lot. The repressilator was actually, it showed up in a book that I recommended. So if you look in the description, there's a couple of books listed there that I think are just good basic like starter books for genetic engineering. And one of them had the repressilator in it, which is how I heard about it. And then I started reading about the repressilator, and then, you know, it's you dig into all the other things that people could do. But a lot of it like, how do I come up with this stuff is either, you know, big bag of weed, or, you know, just, I don't know. I'm a bit of a goofball. I come up with stuff. But also, like, you watch things and you read about things, and I just read constantly. Like, people ask me a lot, like, how do I learn all this stuff? And the answer is I'm constantly reading papers. Like, I will comfortably go through more than a dozen papers a day on a slow day. On a fast day, like, when I'm actually trying to solve something, I will go through way more papers than that. Like, I will comfortably go through 50 or 60 if I have to. So, like, the answer is, how do you become a good biologist? Read constantly. How do you become a good scientist? Read constantly. Always be expanding your knowledge and your tool set, because it just doesn't make sense to not. And I find that the reason that a lot of academics have problems is because they maybe remember some stuff from class, but they don't keep their knowledge fresh. They get stuck in on their problem, and then they stop learning new things, and then you run into issues. So anyway, that is how it is. Okay, what else we got? How would you stop MHC from... Oh, okay. I need to Google this because I don't remember what MHC stands for. Major histocompatibility complex. Yeah, so I mean, if it's repressed, you just express it. Okay. Yeah, so the question was, how do you stop MHC from just showing the immune system now that every cell has some funky virus inside it now? And the answer is there's all kinds of different ways that you can make the immune system competent. So basically one of the ways that you can do this is you take your protein of interest and you make a second copy of it that has the back half of an antibody stuck to it. And this will basically just teach the body that you're not supposed to chew on this, because it generally leaves things that look like antibodies alone. Because antibodies are inherently variable, so they're going to have a bunch of weird code stuck to the end of it. So your body has to have a method for ignoring those. And so you can basically hijack this. There's a bunch of other ways that you can do this. Okay, we also just got a question, which I'll answer really quickly, which is how long until the next Doom Braid video? Next month. We have neurons are en route. We've started filming the stuff. I can actually tell you about this now, which is exciting. We made a deal with Multi-Channel Systems, who are one of the manufacturers of neuron arrays, and they've sent us a full system and a bunch of neuron arrays specifically so that way we can use their system in the next video. So I'm very excited about that. That's going to be just super rad. But yeah, next episode of the Neuron video is coming hopefully next month. But of course, that's actually contingent on how long it takes me to edit the video. But it's basically a sure thing at this point. All the materials have been ordered and have started arriving. All the neurons are going to arrive probably. I was going to say next week, but I'm probably going to be doing a lot of editing next week, so I'm not going to order them so that way they don't go bad, because I just can't be in the lab to tend to them. But yeah, no, Brain Computer is coming very, very soon. Okay, a couple more questions, and then we're going to wrap for the day, because I have to actually, speaking of neuron stuff, I need to go pick up some of the supplies, because they're at the post office. And just one last note about the neuron thing. We're using a ready-made system so that we can test our homemade system. So the plan is episode number 2 is going to be using the commercially made system and testing our homemade arrays. And then episode number 3 is going to be using our own recording system and our own homemade arrays to actually play Doom, or at least get it to solve a maze or something, or any of that kind of stuff. So it is coming. This year is going to be big neurons, big bioenergy. We're growing bones this year. We're probably growing heart tissue this year. There's a lot of really, really, really cool bio stuff coming, which is kind of why we're starting the year off with easy and silly. So this month's video is going to be very silly and very fun. It's goofy. We had a ridiculous idea. I was like, yeah, fuck it, let's do that. But the next couple of videos are going to be really serious and really cool, and I'm so excited. Okay, couple more questions, and then we're gonna wrap this up. Okay, as a curiosity, what is your university background if you want to talk about it? I've talked about this many times before. I dropped out. I started doing a biology program, realized that the program was crap, and the second I had an opportunity to leave, I took it. So yeah, I have no degree. This freaks people out sometimes, but again, how do you do what I do without having a degree? You read an absolute shitload of papers, endlessly, constantly, about everything that you can find. I will I role play as a physicist on YouTube, but I'm not a physics major at all. My math skills are not great. I mean, they're fine. They're not. But I'm not a mathematician. I'm not the greatest math guy. But I'll read papers about asymptotically safe gravity and learning about the nitty gritty of the math and what a renormalization flow is, just because it's neat. It's one of those things. You just gotta just read things that you find interesting. Truly, my educational background can be summarized with I am in love with the universe and figuring out how it works, and I will do literally anything to learn more interesting things about how the universe works. Of course, I mean, I balanced with morality. I'm not gonna, I don't do a lot of experimenting on orphans, because that's not okay. You read a lot of papers, you do a lot of experiments, and you also it's also good to learn about things that are outside of your comfort zone, because you start reading about physics stuff, and then as you do, you go, hey, maybe I can use this for a bio thing or whatever. Or you're reading about a bio thing going, hey, maybe this is a thing I can use in physics or what else. I mean, this is like how the meat laser started, which is like, also, I mean, a lot of the content for the channel comes from like somebody will say something funny, like meat laser, and then we're like, well, I mean, how do you make a meat laser? And then we just sit down and figure it out. So it's like, it's how these things. Okay, we got a couple more questions. When are you getting back to the spider silk? I've talked about this again many times. Which the spider silk project is going dark because we're trying to commercialize it. And until that's done, you're not going to hear about it. But when it's totally finished, you will hear about it. Okay. We got one from Arvel. Do you model the circuits that you make using any computational models? Tuning the concentrations, timing, periodicity, blah, blah, blah. I've not done a lot of work with genetic circuits, so I don't tune them per se. But you could. There are programs specifically for designing genetic circuits. I didn't really get into them in the presentation, but like this exists because people found it really complicated to program these. So that is totally something you can do. But again, the computational modeling is, in my opinion, fucking useless until you actually do the experiment. Like it will give you a rough guesstimation, but until you do the experiment, you're not really going to know how the DNA performs. And like this is something that's actually been really bothering me lately, which is people talking more and more about how like to be a good biologist, you need to be good at coding and computers and stuff. I'm like, no, you don't. You need to have a basic knowledge of those things, but like I would take people who are way like fantastic at lab work over people who are great coders and shit at lab work any day. Because at the end of the day, being a good biologist is actually more important to be able to solve biological problems than it is to be able to code your way through some buttfuckery. Like, the thing is, most things have been solved by people already. Like, the reason we're seeing this big boom of biochemistry is because most of the patents were filed 20 years ago. So they're becoming, like, free to use, and so all of a sudden there's this renaissance of biotech because there's being a ton of money being pumped into it, and also a bunch of patents are expiring. So like, right? Like I'm very, if it disagrees with experiment, it's wrong. So like I am way less likely to try modeling something than I am to just try it, because in the amount of time it's going to take you to model it, you could have just tried it and then found out if it works. Because it's like DNA is expensive, but like your time sitting there coding a thing is costing you just as much as like just ordering the damn thing, and then you can be doing something useful while you're waiting. So like, I mean, again, there are exceptions to this. There's a lot of modeling that can be really helpful. There's a lot of computer stuff that can be really helpful to being a biologist, but like it's not the thing that makes you a good biologist. It's not the thing that's really gonna like advance your work, right? Like of all the people working on like spider silk over the last 20 years, like yeah, they can model it as much as they want, but at the end of the day, they don't have a thing that actually fucking makes good amounts of spider silk. And like that's not a problem you can solve by just thinking it through on the computer. You actually have to like be testing stuff. So okay, let's move on to the next one. And like I say, I'm going to do like 2 or 3, and then I got to wrap it for the day because I really do have other things I got to do, mostly for the Neuron project for the rest of the day. So, why are brains better at, quote, machine learning compared to GPU, which is using much more energy? They're not. I mean, it depends. Like, the thing that makes human brains unique is their ability to extrapolate and come up with new concepts. Like one of the limits with a lot of AI stuff is that it doesn't know how to generalize. Like it doesn't know what to do outside of its training data set. And so you end up being really limited based on what you trained it on. Also, I just fucking hate AI. Like there's a lot of stuff like it's so overpromised and so under-delivers in so many aspects that like I avoid it as much as possible unless it's necessary. Like there are absolutely things that work and are great and are helpful, but most of it sucks. And like a lot of it is just not. It's not good. Okay, what else we got? Can you make a miniature dinosaur from a bird? How hard would it be? I mean, hard, very, but there's people who've attempted it. There's a couple of groups that have been working on trying to make birds more dinosaur-like. It is technically doable. I mean, all the code's in there theoretically. It's just unbelievably difficult, and it's working with whole ass animals. So like the amount of messing with it you're gonna have to do in breeding is just slow. Like a chicken takes a long time to grow up. So like, I mean, admittedly, you can use a different bird, but like, you know, if you're gonna start with something, start with chicken. I mean, better than start with an emu. Those things. I mean, the thing is, birds already are dinosaurs. Like people seem to forget this. Birds are dinosaurs, or most of them are anyway. Like if you look at an emu, that's just a raptor. It's just a raptor. It's just straight up, it's a raptor. But anyway, so yes, you could make dinosaurs if you were committed enough, but it would just cost you an ungodly amount of money. So yes, you could make tiny Jurassic Park, but again, it would probably take you 20 years, and it would be very, very, very expensive. But yeah, you could do it. Again, there's nothing in biology that you couldn't do. You could make cat girls if you wanted. It's just really expensive and extremely unethical. Like, you know, like a lot of stuff would be much easier if we were allowed to do cloning. But like, it's illegal in everywhere, pretty much. So like, eh. Okay. What else we got? Okay, I'm gonna take one more question and then we're gonna wrap it up. Somebody asked, what's your favorite source of papers? Sci-Hub. The answer is always Sci-Hub. It's always Sci-Hub. Like, I mean, there's some alternatives now to Sci-Hub for things that Sci-Hub might not have. But yeah, no, Sci-Hub is the greatest good that that has been done to or for humanity in like 100 years. Like, it's like the invention of like high yielding wheat and the Haber process followed like immediately by that. Like, it's pretty good. It's pretty good. Okay. What else we got? Somebody's asking about a Discord server. Yes, there is a Discord server for patrons and channel members. So if that's something you're interested in, links below. Okay. Okay, last question. Opinion on ZNA enzymes. So for those unfamiliar with this term, ZNA is expanded DNA with extra letters. So normally the nucleotides for DNA are A, G, C, and T. So adenine, guanine, cytosine, and threonine, I think. Tired, whatever. A, G, C, T. But you can make DNA, and you can make enzymes out of DNA, and you can make enzymes out of RNA. In fact, there are many that include at least chunks of RNA or DNA in the protein structure in order to be functional. But you can have a purely RNA-based enzyme or a purely DNA-based enzyme. So if you have a purely DNA-based enzyme, but you add extra letters, so you add like an extra base, this means you have more chemical functionality and variability that you can build into your enzymes. It's very neat. It's very neat. I am not an expert in the slightest in DNA or RNA enzymes. I had a professor who was, and she was an expert in aptamers, which are like sensing molecules made out of either DNA or RNA, usually RNA. Yeah, I mean, they're fascinating, but it's just not something I'm an expert in at all. Because I've looked at them a bit, but it's really like all the XNA stuff is just super weird and niche. So it's not something that like people generally play around with much unless they're like really interested in it. Just because it adds so much complications, because you have to have a special polymerase which is capable of making the XNA in the first place, which that's a whole nightmare. Or you've got to like do it chemically, and then that's a pain. So like it's all very difficult, but it's fascinating. Like, but again, this is what I mean. You could do almost anything with biology if you are sufficiently dedicated. It's just really, really hard. Alrighty, I think I'm gonna wrap it up there for the day. I hope you guys have enjoyed. I've had a lot of fun. Yeah, I'm really excited for the rest of the year. I think we got a lot of videos coming. There's a lot of cool stuff that we've got planned, and it's already in the works. And yeah, definitely keep an eye out for a video hopefully this month, and then the next Neuron video next month. And yeah, I think that's pretty much it. Like I say, links to everything will be down in the description. I'm going to add all of that once this finishes, and then did I say thiamine? Yeah, it's meant to be thiamine anyway. Um, and yeah, so yeah, like I say, I hope you guys have enjoyed